Good evening. I am Madhurima Datta, Zoology and Biology Educational Content Creators. Now's content for Class 12, CBSE, ICSE, other boards and also NEET aspirant students. Unit 1, Reproduction, and Chapter 1, Sexual Reproduction in Plants. And topic is embryo. For the previous slide, please subscribe my YouTube channel Biogenesis by Madhurima for Class 12 playlist. What is embryo? The zygote develops to form the embryo only after some of the endosperm are formed. The embryo develops at the micropyliar end. Micropyliar end means these sections. And embryo are two types, monocot embryo and dicot embryo. At first, we will discuss about the dicot embryo or dicotyledonous embryo. So, first, in dicotyledonous embryo, the zygote divides transversely to form two cells. The cell that lies towards the micropyle is known as the basal or suspensor cells, and the other cell formed towards the chalazal ends, that is the known apical or embryonal cells. The basal cell divides transversely and form two called suspensors. The embryonal cell divides vertically to form two embryonal cells. The two embryonal cells further divide to form four embryonal cells and known as the quadrant stage. And this division of the cell is the mitosis division. Next, the two suspensor cells divides by transverse division and forms a long filament-like structure called as suspensor. Suspensor pushes the developing embryo into the endosperm for nutrition. The micropylar cells of the suspensor swells up and is known as haustorial cells. The cell of suspensors to the embryonal cell is known as hypophysis. The four cells of the quadrant divide to form an 8-cell structure. The four cells of embryo near to the hypophysis is known as the hypobasal cells, and four cells present toward the chalazal is termed epibasal cells. Hypobasal cells give rise to radicle and hypocotyle, and epibasal cells give rise to two cotyledons and plumule. All the 8-cell divides to form the proembryo, that is the globular embryo. The globular embryo divides to form the half-shaped embryo, which form the cotyledons, and later the torpedo-shaped embryo, that is the dicot embryo, has an embryonal axis and two cotyledons. Epicotyle is the embryonal axis above the level of the cotyledons that terminates into the plumule. Hypocotyle is the portion below the level of cotyledons that terminates into the radicle, and the plumule is the future shoot while radicle is the future root. Next, monocotyledonous embryo or monocot embryo. In this monocot embryo, the zygote divides by transverse division and forms two cells. The upper cells towards the chalazal end is known as the embryonal cells, while the lower cell towards the micropyle is known as the basal cells. And this division is also mitosis division. The basal cells increases in the size and forms a vesicular suspensor. The embryonal cells divides and forms a terminal cell known as cotyledon, and the lower cell as known as embryonal cell. A transverse division is embryonal axis cell gives rise to two cells. One cell gives rise to plumule initial and other gives rise to radicle initial. Plumule initial divides to form plumule while radicle initial divides to form radicle. The embryo of monocots possesses only one cotyledon. In Gramini, the cotyledon is known as scutellum, that is present on the lateral side. The embryonal axis has the radicle and root cap enclosed by a sheath called coleryza. The epicotyle has plumule which is covered by the colectile. Now the topic has ended. Thank you everyone.